To understand the plasma membrane and the other membranes fully, we need to understand the phospholipid. We need to know the phospholipid structure. Here's our hydrophilic head. Hydrophilic means water-loving, and it will go into water. It's not going to actually dissolve because it's got these fatty acids attached to it, but it's going to go into the water. And we've got a phosphate portion and a glycerol portion. And it is polar, right? It is polar just the way water is. That's why it will go into the water. The hydrophobic tails, hydrophobic means water-fearing, are lipid. They're like oil. And they don't want to be in water because they're not polar. They're non-polar. It's a non-polar tail. We see one which is saturated, and that means that that carbon chain has every hydrogen on it that it possibly can. So what saturated fatty acid means is that it's got all of those hydrogens on it. Unsaturated means that it's got at least one carbon-carbon double bond. And to have a carbon-carbon double bond, you've got to pull off a couple hydrogens. And that gives the tail different shapes, right? Now, when we take a number of those phospholipid molecules, we can make a membrane. So here we see the bilayer. Bilayer means two layers. There's our fatty acid tails. There are our hydrophilic heads. There's some tails. Here's our second, this is our second layer, right? We got tails facing towards the inside of the membrane. That's into the membrane. We've got our extracellular environment. That means the environment outside of the cell. We would also say interstitial fluid in humans. And so because that's aqueous, aqueous means that it's a water solution, a water environment, those hydrophilic heads will go into the water. Same on the inside of the cell. Our cytoplasm, our cytosol, is aqueous, and the heads will easily line up to go into that water. The hydrophobic tails do not, and they face one another to get away from the aqueous environment. Because we have two layers, we call it a bilayer, and this molecule is called a phospholipid, so that's why we call it a phospholipid bilayer. And remember, we said on the last slide, this is our phosphate group is in the head. Hydrophobic tail, hydrophilic head. All right, arrange tail to tail. Polar heads contact the fluid inside and outside of the cell. All right, here we have a look at our fluid mosaic. Fluid means that these molecules can move around a little bit. Mosaic means that it's made up of these little pieces. So if you've seen those mosaics out there on 99 along Fresno, those are made of small pieces of tile, small pieces of ceramic tile. That's a mosaic. And that's why we use this term fluid mosaic. Right? These are all, they're not glued down the way the tiles on those billboards are. They can move around a little bit. We do have these things other than the phospholipid molecules, including a channel protein. We've got integral proteins that go all the way through. We've got peripheral proteins that don't go all the way through. We've got glycolipids, a lipid with the carbohydrate attached, and glycoproteins, which are proteins with a carbohydrate attached. So some of these are involved in cell-to-cell recognition. Some are involved with, some are receptors for the cell. We also have cholesterol present in the cell membrane, and that is in a healthy person as well. So cell membrane of the cell is a phospholipid bilayer containing many different molecular components, including proteins and cholesterol, some with carbohydrates attached. All right. That's our cell membrane or plasma membrane. So what are some ways that things can get across these membranes? So these membranes are selectively permeable. So if we have molecules that are small and nonpolar, they can get through. Here we are on the outside. So they're going to move from the area of higher concentration to lower concentration. So if we start with more of them on the outside, or we deliver them, say through the blood, they're going to just diffuse in. Nothing is required. So if they're small and nonpolar, they're able to get through those tails, those nonpolar tails, and they're going to fit through, and they're just going to move by diffusion. And we're not going to need any energy, we're not going to need any special channels. It's just going to diffuse through until we have similar concentrations on the outside of the cell and on the inside. So remember, when we're diffusing, we're going to go down the concentration gradient. So these molecules are going to just go right down the concentration gradient from where there's more of them to where there's fewer of them. We can also have facilitated diffusion. So facilitated diffusion is still passive. It does not require any energy, but we might be using a protein channel, right? We may be using a channel that allows those molecules to enter. So channel proteins are less selective than carrier proteins, but they still are going to be limited based on size and the charge of the molecule. Right? And diffusion is still the driving force. We're still going down the concentration gradient. Osmosis is the diffusion of water. Osmosis is the diffusion of water. And remember, in diffusion, the molecule is going to move from the area of higher concentration to lower concentration. So if we're looking at this first beaker, the water is more concentrated on this side. Our solute is more concentrated on this side. But the solutes can't get through this membrane. Water molecule is small and it can get through. So in this case, the water is going to be pulled from this side to that side until we have equal concentration on both sides. Now remember, when you're looking at this concentration, you're looking at this solution, the solute is the thing that's dissolved in the water. The solvent, in this case, is water. The solvent is what the solute is dissolved in. So if we think about other solvents, paint thinner would dissolve paint. Nail polish remover is a solvent that would dissolve the nail polish, right? And so if the solute concentration is higher, the water concentration has to be lower, right? If we're over on this side, we've got more water molecules here than on this side. So don't let that solute concentration get you confused. If we've got more salts and sugars in a given amount of water, we have fewer water molecules, right? Now, another word we need to think about is hypertonic, hypotonic, and isotonic. At this point, they're isotonic, right? They've got the same concentration on both sides. When we start with, this side is hypertonic, this is hypotonic. And here we have some red blood cells in these different concentrations. Here's our isotonic. That means that the solute concentrations in the blood plasma are the same as they are in the red blood cell. This is what we want. We don't want to give somebody an injection of distilled water, which would be hypotonic, right? That would blow up their cells. distilled water would get pulled into the red blood cells and rupture them. We don't want to give them too much salt. It's going to pull the water out of the red blood cells and dry them up. We want isotonic. Iso means same. Hypo means lower. Hyper means more. Now you might have heard these suffixes before. If somebody says, do you hyper, you think of an overactive child. But we can put that hyper in front of lots of different things. So if we say hypertonic, that means that we have a solution around the cells that has a higher solute concentration than the inside of the red blood cells. So it's going to pull water out of the red blood cells and shrivel them. Isotonic, this is what we want. We keep them the same. Hypotonic, that's our water without solutes, and it will tend to move into the red blood cells and cause them to rupture. The sodium-potassium pump is an active mechanism for transport. It's going to use ATP. It's going to use energy, right? It's going to pump sodium out, potassium in, right? But it's going to require energy, and it's going to do it in this particular protein. So when we're going against the concentration gradient, then we're having to use energy, and we call that active transport. All right, we have some other ways of moving things. We've got phagocytosis. In this case, the membrane kind of blebs around a particle and can pull it in. In pinocytosis, that's sipping, right? Cellular sipping. We bleb around some smaller molecules. And then in receptor-mediated endocytosis, we have what we call a ligand, or a binding site, and a messenger. So here we see that this molecule is binding, that's the ligand, it's binding with this receptor and causing that cell membrane to form a vesicle, right? To just bleb in and take a sip of whatever's there. In exocytosis, we're going out. So the processes we just talked about were endo, those were bringing things in. In exocytosis, we're going out. Right? So here comes our vesicle. It touches the membrane. You see that the membranes just kind of bleb onto one another, and the contents of the vesicle are released to the outside environment. In this illustration, we see some pancreatic cells. And these cells produce and secrete enzymes that we use in the digestive system. So these little black dots are secretory vesicles, and they're filled with enzymes, and those are going to be released through exocytosis, right? So all of those are little secretory vesicles. And again, we've got an image of a cell. You want to recognize these organelles, whether they're in this illustration or something similar. The colors are not going to help you. Recognize the shapes and the things about the shapes, right? In mitochondria, we've got those internal membranes. The plasma membrane, of course, is around the outside. The cytoskeleton is represented by these various filaments. Golgi apparatus is really pretty distinct. We've got these flattened membranes that are releasing these vesicles. Vesicles are coming up to the membrane and releasing whatever's there. Vesicle could be quite larger in an animal cell. There's our nucleus, nucleolus, chromatin, rough ER, smooth ER, centrosome. All right. There is a nice view of the rough ER. Take a close look at this transmission EM. You can see the ribosomes. You can see the ribosomes on the rough ER, and that is just closely pressed to the membrane of the nucleus. Okay, and there's a little bit of smooth ER. There's our Golgi apparatus, and again, compare it with the transmission electron micrograph. So this should do a couple things. It should help you appreciate that somebody drew this out for us so that we can see what they're talking about. But also know that it's not exactly the same as it is in the cell. And get in the habit of comparing both. Sometimes in the book it'll show you the cartoon, it might have a diagram, it might have an electron micrograph or a light micrograph. Compare those, make it make sense in your head. So of course the Golgi apparatus is packing and shipping. There's our mitochondria. You can see there we have it in electron micrograph. You can see the internal membranes and the cristae inside. And this is where cellular respiration is occurring.